All right. Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us the 80th seminars. And I call it a Stanford University. I came back to Germany again to meet people and compete for a crazy idea prize awarded by German funding agency called Sprint. It was so fun to meet people with crazy and disruptive ideas that actually, one of the speakers today, Drew, actually had and still has, for 20 years. And then I was also meeting a lot of people with the crazy ideas today and yesterday during that the event. I want to thank the organizers, including Drs. Patrick Rose and Scott Wolfer, who gave their amazing talks for my seminar series recently as well. Okay, it is my tremendous honor to introduce very briefly, but I'm not sure I can do so, our pioneer speaker, Professor Drew Endy. He does not need any introduction because we all know him. He is a professor at Stanford University. I have known him for almost 20 years since he was a professor at MIT, where he studied iGEM activity, of course, and participate in the weekly MIT synthetic biology workshop group, a working group workshop, where I met him regularly to discuss the future of synthetic biology. He has contributed to establishing the synthetic biology field in early 2005 when he published one excellent review paper in Nature titled Foundations for Engineering Biology, which I use still for my synthetic biology course for more than 10 years. Drew, I truly thank you for your time and for your contribution to the scientific society and establishing the exciting field many enjoy right now or last 20 years, and the virtual podium is all yours. And thank you so much again.
Thank you so much, Professor Moon. I'm super grateful to be here with you and with Professor Natalie Farny, who I'm more excited to hear about than anything I have to say. Although we're 20 years into the modern version of synthetic biology, for me, it really feels like we're at a starting line. Because what's happened over the last 20 years is we've learned and are continuing to learn what the ancient meaning of the word synthesis really is all about. A lot of times when I grew up in synthetic biology, people would ask the question, is that what we should be calling it? Should we be calling it synthetic biology or engineering biology or genetic engineering or shiny happy biology? If we called it shiny happy biology, then there'd be nothing to worry about, right? It'd just all be shiny happy. And it's a really good question to ask. And as I've gotten older and explored the field more, still coming back to a new starting line, as much as I love biology, I think the more important word is synthetic, is synthesis. And whatever language you love the most and its origins, go deep back. I dare you, in a friendly way. I really encourage you to go back in the language that you're most in love with and explore the ancient meaning of the word synthesis in your favorite language. For example, it's not the best example, but it's one. If you go back into the Greek, you find the word synthesis. The Y is replaced with a U. And what is the meaning of that? It's composition, a putting together. It's so simple. It's so brilliant and powerful. It's like a putting together. And when you put that word in front of biology, it's like has so many meanings. Of course, it has meaning at the molecular level, like we're a putting together the molecules. but it has meaning at the cellular level, at the ecological level, at the cultural level. It's just so profoundly interesting. And so as I've been playing and exploring and enjoying the fruits of synthesis, so to speak, I've really come to appreciate that part of it, the putting together of things. Now, there's another thing that makes me feel like we're at a starting line. And it's really a couple things coming together. The first is, it feels to me like we've now been able to just barely diagnose the immaturity of our culture broadly with respect to biotechnology and putting biotechnology together. For 50 years, of course, we've had genetic engineering, but the pressures on genetic engineering have been overwhelming from a utilitarian perspective. Like, do the useful thing right now to mitigate suffering. It's totally appropriate. But when you drive something only via utility, you don't have the more mature filling in of broader societal engagement. And I don't just mean understanding. I mean an acknowledgement of fear. I mean an ability to play and learn what something could be or might be and what we might wish for. And so one of the things that's very exciting, this partnership with design in the arts and synthesis in biology, is finally bringing constructive critique back in a way that's impacting the natural scientists and the engineers. And wow, that's going to get us to a biotic society, or I should say societies, plural. So that's amazing. The second thing that feels amazing to me and a fresh starting line is I can string the following words together, but more importantly, so can other people. Biology is a general purpose technology. Biology is a GPT. I don't mean chat GPT. Biology is a general purpose technology. Anything that we can encode via DNA, we have a chance of growing. And we're familiar with the industrialization of it through production of medicine and food and materials, but that's really not what it's about. You know, that's 19th century metaphor and practice and concentrated deployment. Did you know the Semiconductor Research Corporation in 2018 published a roadmap for synthetic biology? Let me back up. The Semiconductor Research Corporation, operating at the intersection of biology and materials. And the first thing they want to do is put data into DNA. We've seen books going into DNA over the last decade, but they want to put like the Library of Congress into DNA. Oh, abiotic DNA. And of course, now we have the DNA Data Storage Alliance. But you know, then they have steps 2, 3, 4, 5. Step 5 is we're growing computers. We're using bottom-up organic templating and self-assembling of organic materials to position the atoms in hardware, right? You know, it's fantastical because we're not close to being able to do that, although there's hints of how we go about it. Really interesting pioneering work all over the place, Paul Rothman and others. But like, what if we have a general purpose technology? What do we need to do to unlock that? And when you ask and answer the question, what do we need to do to unlock biology as a GPT? It's really interesting. We don't have to do a million things. We only have to do like 7 things. A small integer number of strategic programs would get us that. Okay, and then that gets me to the third thing. I think that by or before the year 2050, if we are as amazing as each of us can be, and all of us together, we can help everyone on this planet get to a flourishing planet. And I'll just, because I'm an engineer, I'll round up and make the math simple. Up to 10 billion Homo sapiens independently operating on this place in partnership with the rest of life on Earth without trashing it. I actually believe it's physically possible to realize that type of practical transformation on a planetary scale because of what's happening in the renewable energy sector, because of what biology represents as a platform for taking energy and organizing matter, and because of what we can do with the network. Our greatest challenge, of course, is not only bringing forward the biotechnological platforms and solutions and scaling that, like iGEM's going to have to become at least 1,000 times bigger. But we're also going to have to figure out how to navigate and renew our cultural, political, economic systems to be compatible with abundance and flourishing. But honestly, the starting line I feel as an engineer that's most practically exciting for me is this seems, when you do the math or you work it out on the whiteboard, physically possible, right? And now it's a task of turning that into both R&D programs, but a cultural movement that makes this real. I just want to pause there. I'm much more excited to hear from Professor Farny today. And I want to highlight some things from afar that I find amazing about you. And just two things. The first is, from what I can read, and I don't know that I appreciated this, you have an undergraduate degree both in biology and philosophy. Is that right? Yeah. So
yes, I do.
that's amazing. And I just wish there were more folks like with those dual perspectives, like coming from points of origin that let us see things in different ways. And then the second, your research is very inspiring for me because it's combining what biology can do in nature and bringing it not only to people health, but finding this relationship between planet health and people health. By understanding what's going on in nature, by developing the sensors, by having empathy for the give and get, right? So much of what happens in engineering puts the human, the Homo sapien, in this isolated box and tries to keep nature away. And that's not how we're going to get to it. So just like really thrilled with what you're up to in pioneering. Professor Moon, maybe I'll turn it back to you for a more formal introduction.
So thank you so much. Oh my goodness, you always make me think, and then you make my memory about synthetic biology refresh, and then I need to do it again, and then I will think about what you said over the weekend. That's absolutely brilliant, and always thank you for your, pioneering, visionary, remark and talks. And that's why it's my great honor again to have you today. Thank you so much. Okay, thank you. Okay, so now the main speaker of today with a longer introduction, Dr. Natalie Farny, on a BS in biology and philosophy, and Drew already know about that, from Boston College, and the PhD in cell and developmental biology from Harvard University in Dr. Pam Silver's lab, where of course she studied mRNA nuclear export pathways. From there, she completed her postdoctoral work in Dr. Richter's lab at UMass Chan Medical School, where she studied mRNA metabolism in the brain. It's interesting, she working on brain. Also, she did, philosophy, degree, bachelor degree. She first became engaged with the synthetic biology as a iGEM team mentor, then went on to be a judge and served as a co-chair of the iGEM measurement committee. I think that this iGEM is an interesting connection among three of us because, Professor Drew Endy started it, and I have been involved in that competition for more than 10 years, and now as a iGEM startup advisor, and then Dr. Farny also heavily involved, in that competition. She is currently an assistant professor of biology and biotechnology at Worcester Polytechnic Institute. Her lab studies the relationship between cells and their environments in many contexts. She is particularly interested in the effect of soil and water contaminant on cells and on the application of synthetic biology to understanding and remediating environmental contaminant. She is the recipient of US EPA Early Career Award and also WPI Board of Trustees Award for Outstanding Teaching. Her lab is funded by multiple agencies, NIH, NSF, EPA, and DARPA, and ARL. I believe she is a true rising star, And she and I also organizing an international conference on microbiome engineering, where she showed a true leadership I normally do not see from junior faculty. I want to say that if you are a potential tenure letter writer, you will want to write your strongest letter. And Natalie, thank you so much for your time today, and please take it away. It's all yours now. Thank you.
Wow, thank you. Thank you so much, Taesuk. That's an incredible introduction. And thank you so much, Professor Endy, for your introduction as well. I really felt that your remarks really resonated, and hopefully I touched upon some of those in my talk today. So I will just share my screen now. Are we all good? Okay. Great. So thanks. It's really a delight to be here and to be talking to you today about the work that's going on in my lab. The title of my talk today is Survival and Function of Engineered Organisms in Model Soil Environments. But I'm going to touch upon some things that are a little bit beyond that today as well. Just in case you're not familiar with Worcester Polytechnic Institute, we're in Worcester, Massachusetts. We're about an hour west of Boston. So this is our beautiful campus. And I've been at Worcester Polytech for quite some time. And I like to show this slide just because I've had somewhat of a nonlinear path to becoming an assistant professor. And I think that it's important to share and to normalize the idea that there are multiple ways to get to your professional goals. And so I won't go through this entire slide, but sort of the point that I have here is that there were a lot of different options and numerous paths not taken along the way to get from my undergraduate degree to the position that I'm in now. And I'm always happy to talk to other folks out there about these kinds of issues. So if you are a student or a postdoc and you're thinking about career transitions and making tough decisions in your career, and any of this is something that you want to talk about, please ask me. I'm happy to answer questions and talk about this with you. And the other thing I like to point out is really the role of many amazing mentors that I've had along the way that have intervened at many of these arrows to help guide me, but also not only to help guide me, but to sort of break down doors for me when that needed to happen. And so I'm really grateful and like to acknowledge that at the start as well. So my lab works on sort of this intersection of synthetic biology and the environment. So we're really interested in the effects that the environment can have on cells, and also the ways that we can engineer cells to have effects on their environment. And the lab is really broad. So we have a number of different interests, some of which stem from my training and my history in mRNA translational control. So we do still do some work on cellular stress response and the formation of stress granules in response to environmental exposures like bisphenols and heavy metals like arsenite. We're particularly interested in the role of physiologically relevant types of stresses on the formation of stress granules and biomolecular condensates. So we do some work on that. We do some work on biosensing and bioremediation in real world soil and water environments. So I'm going to be talking mostly about this today, understanding how engineered organisms function in real world soil environments or in models of soil. We're also doing some work through a nice collaboration with Jagan Srinivasan's lab at WPI on chelation therapies for metal toxicity using C. elegans as a model. And so we're really excited about this work, and it should be coming out really soon. And then finally, as was mentioned in the introduction, I've been engaged in iGEM for a long time, and as co-chair of the iGEM measurement committee was heavily engaged in the research surrounding the interlab studies and standardization of biological measurements. And so you'll see that influence of measurement sort of sprinkled throughout my talk today. And again, if you have interest in any of these other projects that I'm not going to cover today, please do reach out. I'm happy to talk to you about any of those things. But I'm really going to focus today over here on soil. And so I really appreciate what Drew was saying about thinking about humans in the context of the environment and as a part of a broader ecosystem that we need to consider. And that there are ways still for biology and synthetic biology in particular to intervene and create a world where humans can coexist with nature without destroying it. And so the particular issue that I'm going to focus on today is soil. There are a number of global threats to soil. You may have heard this statistic before, but much of our planet's diversity is found in our soils. There are more organisms in 1 gram of soil than there are humans on this earth right now. And so there's a lot to be learned from our soils, but also a lot to be lost if we don't become responsible stewards. So there's a number of different threats: unsustainable soil management practices, pollution, so effects of contaminants. We're losing farmlands to contaminated soils, to compression, to erosion in particular. So all of these are important threats, and we need to start to think about how we're going to remediate those threats. Of course, the soil also serves as an important carbon sink as well. And so if we can increase the pool of organic mass and biomass in our soils, we can reduce the amount of CO2 that is released into our atmosphere. And so there's a lot at stake here when we're talking about soil health. And there are a number of companies, synthetic biology companies in this space, who are working on solutions to global soil threats. So how did I get involved with soil? So as you heard from my introduction, and also in that first slide that I showed you, I don't really have training in soil biology or microbial ecology of any kind. But I got engaged through this kind of crazy DARPA program that I was a part of with my collaborator at WPI, Eric Young. And so we answered a call from DARPA through the BioReporters program to see whether or not it was possible for bacteria to send an inducible signal through a meter of soil to indicate the presence of explosives underground. And so the idea was you might have an engineered species that's able to detect TNT and be able to propagate some signal to the surface. And we were looking at fluorescent signals. And so we put together a series of gene circuits, which were sort of partially functional. But I think, and I'm not going to really talk a lot about that today, because what I really want to talk about is how this project sort of humbled us, at least me in particular, and made me understand really a lot of the assumptions that were underlying the idea that this was even possible, that either weren't accurate or weren't well enough understood for us to be able to really do it. And so I'd say, can you send a signal through a meter of soil? It's a definite maybe. There are a number of important assumptions there that sort of undermined our process. And one of those was that the assumption that a synthetic gene circuit would be functional in soil. So when you put it into an organism and you put that organism into the soil, that actual circuit would still be inducible and would still work. And so we found, in fact, that that's not the case for most cases. And also that synthetic microbes or engineered microbes would live long enough in their target environment to actually do their job. And again, we found that wasn't actually the case, and I'll show you some data for that. So we were able to get this to work, and Eric has given one of these SynBYSS seminars before, and so you can look up his video if you want to see the data on that. But if you remember his talk from before, we were able to show that you could propagate a signal over a distance of about 10 centimeters through a column of soil if the soil was sterile, and you could only propagate it using homoserolactones as a signaling molecule. And so we tried a number of different types of circuits to induce here. So the target we were trying to be able to detect was TNT, but we never were able to get that to work. nor in fact any of these other signals that we were trying to get to work. So we could get quorum sensing to work using HSLs. However, we were not able to trigger the circuits using any other type of signals. We weren't able to kick off this feed forward circuit by detecting something in the environment that we wanted to detect. And so what that tells us is there's something fundamentally that we're missing about our understanding of how organisms regulate gene expression in the real world environment of the soil. There are other challenges that were part of this project as well. And one of that was just the sort of boring fact of sample processing. So in order to make any of the measurements that I just described, you have to extract your soil sample. We added PBS and mix it up and pellet the large pieces and then filter it out. And then you could load it into the flow cytometer. And so in terms of iterating, like really trying to implement a design, build, test, learn cycle, this was really very cumbersome. And so we thought there just has to be a better way to do this. And so what I'm going to share with you today are the insights that have come from that initial DARPA program that have really spurred a whole new area of research for me and for my group. And so that first challenge is measuring the survival and persistence of an engineered organism in a complex target soil environment. So how do we even know whether this organism is living? And then we can try to modify that organism to modulate its survival in its target environment. So that's our first challenge. And the second challenge is understanding the function of synthetic gene circuits within the soil, when they're in the soil, using our chassis organism, which is Pseudomonas putida. So I will briefly introduce now our model organism, Pseudomonas putida. It's been used in a lot of different contexts. It's been used for bioremediation and bioprocessing. There are a number of excellent genetic tools available in Putida. The one that we are using comes from Adam Guss's lab at Oak Ridge, and they very kindly shared their strains with us. There's a recombinase system that allows for direct integration into the genome of all the circuits that we are testing, and so that's been really handy, and they've been extremely generous with their resources there. And so Pseudomonas putida is a native soil organism. And so given that there are great tools available and a lot of uses, and so relatively more literature than other soil organisms, we're using this as our model organism for the soil environment. And as a sample engineered organism, right now we are using a strain that has the recombinase system and has integrated the TD tomato fluorescent protein as our sort of engineered strain. So this engineered strain that we're testing right now doesn't have an environmental sort of target function, but the idea would be to swap that out in the future for target functions. So this is the organism that we're putting into the experiments that I'm about to describe. So the issue of survival in the soil environment. So we observed in those initial experiments from the DARPA program that our engineered organism, we would put it into a soil column and then we couldn't find it again in several days. And so we had to use sterilized soil. And so just to get sort of a number on that, we did a simple colony forming unit assay where we add our engineered model P. putida to both autoclaved sterilized soils and non-sterile soils. We take samples over time, filter extract them, and then plate these out with serial dilutions to count colony forming units. And so what we find is that in sterile soil, Putida do have a log phase of growth over the first 2 days. This is days. And so it's not that they can't grow in soil. They have what they need metabolically to actually be able to grow. But the competition is what is killing them. So we find that between day 4 and day 7, there's sort of a population crash. And we saw this over and over again, that between day 4 and day 7, or day 4 and day 6 or so, that we would lose most of our engineered Putida, but that you would always find a few colonies. There was sort of this long-term low-level persistence. And so if you want to remediate contaminants in the soil environment, this pattern of behavior, of survival, rather, is maybe not what you want, right? That might not be ideal for your purposes. Maybe it takes more than 4 days for you to remediate what you want to remediate. And beside the fact that colony forming unit assays are really yucky, I mean, they just take so much resources and so much time. Is there something else we can do? So the next thing we tried was to try to measure the Putida in the soil by flow cytometry. So we basically did the same thing. We'd had autoclaved, sterilized and non-sterile soils. We add our Putida, take extracts over time, and then measure by flow cytometry to just try to look for red fluorescence. And we see a very similar pattern. So we see that there is this log growth phase in the sterilized soil, which is in the blue line here. And that we do have this crash again between day 4 and day 7, where the engineered bacteria are no longer detectable. But the sample processing here really wasn't any better. It was easier to do flow than to do colony forming units. But the processing here was really not any better. And it's still the kind of sample prep that clogs your flow cytometer and creates headaches. And after day 7, really weren't able, amongst the background of soil particulate, to discern the persistence that we were able to see on the CFU assays. And so this wasn't really great either. And again, physically taking samples out of dishes full of soil is not exactly what's going to allow us to sort of iterate on our design processes really rapidly and make a lot of progress. And so there just had to be a better way. So we hit the literature and we looked for other ways that people are modeling soil environments. And what we found was this really interesting model by Volker Rosa's lab at South Dakota State University, which he has called soil extracted solubilized organic matter, or SYSOM. So the idea here is that you take your different types of soil, you make sort of like a mud pie situation out of this, and then through a series of filtration steps, you end up with versions of the soil that represent the solubilized organic matter, which can be used like any other media to grow your cells. And so here's our 3 different starting soils and 3 different SYSOMs that came from those different starting soils. And so you can really appreciate the differences in the organic content that come from the different starting materials. And so we thought this would be great. Let's give it a try. So you may be wondering what's actually in this SYSOM. So what is SYSOM and what's in it? And what it is depends on the sample. And so, sorry, back to this slide, you can really appreciate again that they look very different depending on where the soil comes from and what its organic content and its mineral content is. And so, but in general, there are a number of different components that will support microbial growth, organic acids, amino acids. There's all kinds of sugars, fatty acids, and as well as metal ions. And the Brosel lab was able to show that there are a number of different environmental isolates that grow reasonably well in these SYSOM extracts. They also did some really nice work looking at bacillus strains and transition of bacillus strains from their growth phase to their spore forming phase, and showed that the SYSOM was a much better model for that spore forming activity than something like LB. So this is great. So we gave it a try. And so what we did is we create this SYSOM, but then we decided to sort of add another layer to this, because the other part of what was concerning us about the soil models that we felt hadn't really been addressed is the issue of the sterility, right? The competition of your engineered organism with the native microbiome. And so we asked, is there a way using CSOM for us to capture that that interaction, right? Between the engineered organism and the microbiome. And so we thought, well, what if we just left some of the microbiome behind? So we, instead of just doing this 0.2 micron filter to essentially create a sterilized media-like extract, we tried a process that we used 11 micron filtration, which left much of the microbiome intact. And so we have both now sterile and non-sterile liquid soil models that we could use to test the survival and persistence of our engineered member. And this idea of building microbial communities is, of course, not new. There are many people building microbial communities. One good example that's related to soil and environmental health is the simplified community model by Roberto Coulter's lab at Harvard, where they were able to recreate a maize root community from 7 members, one of which is Pseudomonas fetida. And we're able to show really nicely that the introduction of this C7 community, these 7 members, helped to stave off the fungal colonization of the roots by a fungal pathogen. And so there are a number of these simplified bacterial communities in the literature, but the restriction here is that they have to be culturable members, right? So in order to actually build this community here, in these ratios, you have to be able to culture each of the members in isolation. And so that's while these are important models and have contributed a lot to our understanding, they don't recapitulate the given, the established microbial community that might be in your target environment. And again, there's expectation that that target environment that target microbial community is going to be different from environment to environment. So wherever you are, whatever your contaminant situation is, that microbial community is going to be different. And so is there a way to sort of capture the microbial community that you're targeting, rather than trying to sort of guess which culturable members more or less represent what you're trying to do? And so again, back to our model, we thought if we take soil from our target environment and we apply this 11 micron filtration, then some representation of the soil microbial community would be left behind. And the question then is how stable is that, and how good of a representation might a model like this be? And so to answer that question, we did 16S DNA sequencing to look at the soil microbial community. So we took a sample directly from the soil, took DNA extracts at day 0 and day 14. Then we made our 11 micron CSOM filtered extract, and then again took DNA samples at day 0 and day 14. And so what you can see here, we're looking at total class abundance represented in our DNA samples. So the 2 columns on the left are the solid soil samples. And you can see they're pretty similar. There's not a lot of change that goes on in a given dish over this short time period. And then the 2 columns on the right here are CSOM extracted microbial communities. This is day 0 and day 14. And so what we're observing a number of things that we think are really important. So first of all, there are a lot of non-culturable members that are maintained over this 14 days that wouldn't be able to be cultured otherwise. So we are able to maintain some representation of the microbial community. But interestingly, what we find is as we sort of let the microbial community settle over the 14-day time course, the day 14 samples become more like the solid soil samples than the initial extraction. And so this is really early data, but what we think might be happening is that there's if you extract the microbial community and then give it time to rebalance itself, what you end up with is a liquid version that's really a very faithful representation, probably one of the most faithful representations of a starting solid soil microbial community that has yet been documented. And so we're really excited about this model because then it allows us in a liquid format, which is amenable to high throughput types of liquid handling, and to recreate an authentic soil microbial community. And so then the first question is, well, how does our engineered member do when you put it into this liquid soil extract? And so we're delighted when we saw that in the sterilized liquid soil extract, we have more or less stable population of our engineered member. So this is the percent of the cells that are expressing TD tomato of the total detectable cells. And so here we start, and our population maintains relatively stable over the 14 days. And then we introduce at high concentration our engineered member into our non-sterile soil community. And we see again the same crash between day 4 and day 7. But now we're able to detect this long-term persistence. And we've taken these experiments now out to 28 days, and I don't have the data published here, just processed here just yet. But more or less, we're recapitulating this behavior that we saw in the solid colony-forming unit assay, but in a way that allows us to very rapidly, using automated liquid handling, to detect this this decline in population as a result of competition from the existing soil microbial community. So this is really great because this gives us a model now where we can actually test our interventions to modulate this. So if we want to kill off our community sooner, or if we want it to last longer, we have a really easy and flexible platform now that we can use to do those experiments. So what's next for our model? This is really sort of early work, but as I mentioned, we have a couple of important questions that we'll be asking over the next year or two using this model. And so the first is the effect of environmental variables. So as I mentioned at the beginning, we're really interested in environmental contaminants. And if you're putting in an engineered P. putida species that's meant to remediate some kind of contaminant, what kind of effects does that contaminant have on the stability of the microbiome and changes of the microbiome, on the stability, survival, and persistence of the engineered member, and so on. And so we can do things like introduce different concentrations of our engineered member, different concentrations of target and environmental contaminants that we might want to mitigate. We could also introduce culturable members that may have the ability to modulate the survival or persistence of the engineered member. And so we saw in that work from Roberto Coulter's lab that these members really work together to support the health and the development of the maize roots. And that if you remove some of these members, then there's an imbalance in the other 6 remaining members. And so what that says is that perhaps introduction of some culturable species into the microbial community might change the survival or persistence of our engineered member in ways that would be difficult to detect, again, using a solid soil model. And so those are the types of experiments that we'll be able to do now in high throughput using automated liquid handling and flow cytometry. And then other important questions that we want to ask, which I'm going to start to talk about today. This again is really early days for this work in my lab, but I'll tell you how far we've gotten so far, is testing synthetic gene circuits, testing and optimizing synthetic gene circuits for function in soil environments, as well as understanding both genetic and epigenetic regulation of gene expression in the soil environment, which is really understudied. So we're not even sure in a native putida, which may be in the soil, what are the genetic and epigenetic changes that happen that permit survival and persistence in the soil. So that's really an open question that we hope to address and then hope to be able to leverage to build better circuits. So as I just mentioned, this is sort of the second challenge that we're moving into now. The idea of synthetic gene circuit function in soil and soil organisms. And so the first part of this story is really a targeted approach that was taken on by a really talented postdoc in my group, Felipe Carrillo. And so he just made a simple observation that the optimized -10 and -35 sites that correspond to sigma 70 were not represented in the most widely utilized versions of LAC and TET promoters. And so there was some evidence in the literature that LAC and TET induction in Putida was really suboptimal for some reasons that weren't really clear. So Felipe had the idea to just change out these -10 and -35 binding sites to match the optimized sigma 70 binding sites, add a strong RBS that would be the strongest ones that we knew about for P. Putida, and to see if this changed the gene expression, the induction behavior of these circuits in Putida. And this is recently published work if you'd like to see the whole story. But I'm just going to focus today on the TET promoters, but we have similar data for the LAC promoters as well. The assays that I'm going to show you next, we're switching to a fluorescent protein, which is called M cardinal. This is a far red fluorescent protein. One of the reasons why we're switching is for these experiments is because we realized that autofluorescence is a significant problem when you want to do really quantitative measurements of promoter function. So this is a problem in soil. These are spectral sweeps of different types of soils and soil extracts, and you can see high autofluorescence in the green wavelengths, which are much reduced the further red you get. We also found, even just in LB, that when you look at wild type P. Putida under the green excitation and emission wavelengths, and then this is in the green here is P. Putida that are expressing SFGFP, that the signal to background ratio is really pretty small. So you're losing a lot of your dynamic range to autofluorescence in the green wavelengths when you use GFP. And you can see the significant difference when you make these same measurements. Again, Putida wild type, they have no fluorescent protein, and then Putida with M cardinal, that you get a significantly enhanced signal to background ratio and expands your dynamic range significantly. And so you can see that the direct comparison of these two graphs here in this third graph. So these are all, sort of hearkening to my fondness for measurements and really trying to make sure that we're getting the most out of the measurements that we're making of these promoter systems. So using our alternative TET promoter architectures, the original TET promoter architecture that we started with is the version 1. So you see that in the red triangles here. Felipe did this really carefully and methodically. He started with constitutive versions, so in which case the regulator TET-R is not included in the constitutive versions on the left. Then measurements under the repressed state and the induced state. And what you're able to see here as a reference is the TAC-I promoter, which is one of the strongest that was known at the time in Putida. And so we see that the original version of the TET promoter actually, as a constitutive promoter, is very strong, but as an inducible promoter, really only reaches half of its potential dynamic range. But making the adjustments that Felipe made, this second version is able to achieve its full dynamic range, in Putida, even though it as a constitutive promoter doesn't behave very strongly. And so sometimes the predictions that you might make based on just having inducible data or just having constitutive data are not showing you the whole picture of what's going on with your promoters. Incidentally, we did the same types of experiments in V nitrogens as well. This is a fast replicating marine bacterium. It's of interest for remediation in high salt marine environments. And we're able to make significant improvements to inducible gene circuits using tetracycline in B nitrogens as well. And you can build other things from these as well. So Felipe built the beta carotene production pathway using these, so controlling lycopene formation by controlling the TET promoter, and then conversion of lycopene to beta carotene on the LAC promoters, and was able to show that actually both P. putida and V nitrogens were able to make more beta carotene than E. coli, and that because E. coli promoters are so leaky, that essentially, even though you see gene induction when you use fluorescent reporters, the sort of practical behavior of these LAC and TET promoters in E. coli is more constitutive. And so again, a lot of this is either published in our previous paper or in a manuscript that's in preparation. And so this was sort of a targeted approach to improving constitutive and inducible gene expression for P. putida, and sort of looking at it in a couple of different contexts. So this work is really fantastic and definitely expands the genetic toolbox, especially for folks who want to use more environmentally relevant types of organisms like putida and V nitrogens. And so that's really fantastic. However, when we did try to use these in soil or CSOM, they did not induce. So we're still missing something. It's not just about trying to boost the signal or making it more efficient under laboratory conditions. There's something fundamental that we're missing about the way that genes are expressed in the soil that we haven't captured in this system. And so to sort of get at that, then I think we have to take an unbiased approach. And while we were generating the data sets that we wanted for ourselves, we decided to start with a comparative transcriptomics, or sort of meta-transcriptomic approach using published data sets. And this is a combination of work from Stokely, Gabby, and Sue Chin in my lab. We started with a really simple question. If we look across different P. putida transcriptomics data sets, can we identify genes that consistently have high expression but low fold change across a variety of environmental conditions? So the logic here is that what we really want, if we want to build really robust synthetic gene circuits to function in soil environments, what we want are gene circuits and regulatory parts that are going to be relatively agnostic to the environment. So they don't care if it's high salt or low salt or soil or weird carbon sources, that they're just going to be expressed relatively stably all the time. And that if we could identify genes like that and understand how that stability is achieved, that we could work those into more robust synthetic circuits. And so Stokely and then Gabby started to work on this question and came up with a list of genes that most of which are things you might guess. So you might guess that things like ribosomal protein genes or RPOD, Sigma 70, these are things that if you're going to survive, you might need all the time regardless. And so we did see a strong representation of these types of genes. But the gene that came out on top time and again was this odd gene that we hadn't really thought about before. We didn't initially know what it did. But what we noticed is how large it was. This is by far the single largest gene in the Pseudomonas putida genome. It's over 8 kb, and the next closest gene is less than half the size. And so that just struck us as sort of odd. And we sort of looked into it further and found that, in fact, there was one other study that looked at this gene, which is called BLAP-A. And it is an outer outer membrane protein. It has to do with biofilm formation, surface adhesion. And what's particularly interesting about this gene, in addition to the fact that the coding region is so large, is that the promoter region is unusually complex for a prokaryotic organism. There are 8 putative transcriptional start sites upstream of LAP-A. And that was documented in this article here. And so we started to think, is there a way that are we utilizing this promoter structure complexity as a way of ensuring stability, right? So, one way to make sure that your gene is expressed all the time is by having one super strong promoter that just works all the time. But another way to make sure that your gene is expressed robustly all the time is to have a whole series of a variety of different types of promoters, such that under any given condition, you'd have transcription, and that you could basically switch the way that you use this entire regulatory region depending on the environmental conditions. And so in this paper, the authors here show the 2 conditions that they looked at were the difference between exponential phase and stationary phase growth, but they did see an increase in transcription in stationary phase. And so we just asked then, if we go back to these data sets, so all these different data sets were comparing different conditions. So in this case, there's 4 different carbon sources, and here's a difference between control versus high zinc conditions, for example. If we go back to some of these data sets, do we see a difference in promoter usage for this particular gene? And so again, this is work by Siqin Li in my lab. So Siqin looked at a couple of different data sets and mapped these to the putative transcriptional start sites, which are indicated here by the dashed lines. And so she did see some differences that, in this case, this is treated with the antibiotic imipenem, and that there was this increase, these purple and pink traces, increase in transcription in response to short amounts of imipenem, and then decreasing again after longer exposures. Similarly, there was an increase in transcription in response to high zinc conditions, and then also indications that some other transcriptional start sites start to be utilized in response to these environmental changes. So we thought, okay, that's really interesting. So that was sort of what we were working on while we were waiting to get our own data sets, which we did recently get. So Stokely in my lab generated RNA-seq data sets that compared P. putida in log versus stationary phase in both LB and CSOM, so that we could really directly measure, what are the changes happening in CSOM. And so we would see a lot of different things going up and going down. They seem to sort of be almost inverted. But what I noticed at the top of both lists there are a number of genes which are sort of high, but then stay on or go higher in the CSOM condition. So this is LB and CSOM in log phase, and then LB versus CSOM in stationary phase. And so then going back to the example of the LAP-A gene, we just asked, well, does that play itself out in terms of the regulation of LAP-A? And so we find that it does. So in log phase growth, there's high expression from these more proximal promoters, and lower expression from the CSOM. But the pattern completely flips when the cells enter stationary phase. So in stationary phase, in the LB condition, you have very low expression and much higher expression in the CSOM condition. So what we're taking from this is that our initial approach of sort of taking a broader metagenomic approach to identifying genes that might be useful to incorporate into synthetic gene circuits was really validated then when we actually analyzed our own LB versus CSOM data. So I'm going to wrap it up here. We have a lot more to do, of course. We're going to actually build all the synthetic gene circuits using the parts that we've identified. But just to summarize what we've achieved so far with respect to these two challenges. So with the first challenge of survival and measurement of P. putida in soil environments, we created a seesaw model that was able to recapitulate the survival and persistence dynamics of our engineered P. putida in target soil environments, which maintains the microbiome relatively intact, is one of the best, we think, one of the best representations of a microbiome model that's really amenable to manipulation in the lab. And it's really adaptable for high throughput assessment of environmental variables, screening of mutants, looking at transcriptomics, which I just showed you, optimizing genetic circuits, making improvements to our chassis, and so many other things. And so really, this is just the beginning for us in terms of the exciting things that we'd like to do with this model. And then in terms of challenge 2, we had some success with our targeted approach and developed new lac-intent architectures for driving gene expression, including toxic gene expression and biosynthetic pathways, both for P. putida and for V nitrogens. We're happy to share these reagents if anyone is interested. But then also had some success as well with our unbiased approach of comparative transcriptomics to identify candidate regulatory parts for development, for further development in soil applications. And we'll be continuing to work on that in the future. Of course, all of this is not possible without the fantastic people who work in my lab every day and work so very hard. Particular thanks for the data that was shown here to Felipe, Lexi, Stokely, Gabby, and Sichin, who really generated all the data that you saw here today. This is my full group. We're a big group, lots of undergraduates, and my collaborators, Eric Young, and also Adam Gust, who's very generous with his strains and reagents. And then, of course, my funding sources. Most of the work that you saw today was funded by the Early Career Award that I got from the EPA. So I'm very grateful for that support. And I just would like to wrap up with a shameless plug, as Tasek mentioned at the very beginning. I am co-chairing with Ben Rubin and Brady Kress of Berkeley, the 6th International Conference on Microbiome Engineering, which will be taking place at Berkeley, December 8th through 10th. So abstract submission is open. We have a fantastic lineup of speakers. If you're interested in microbiomes in all contexts, so soil, industrial, human, any type of microbiome, please do join us. We are going to have an unprecedented, I hope, an unprecedented amount of financial support available for early career folks, students, postdocs, and assistant professors, and underrepresented minority scientists. So, please come and please stay tuned for more information about financial support, and definitely reach out to me if you have any questions about that. So thank you very much for your attention. I appreciate it.
Wow. Oh my goodness. It's amazing. I believe, Drew Endy should leave soon. So, but he has a lot of questions. So let's hear from him and then, so please go ahead, please.
Natalie, thanks so much for your amazing talk. And I know you're going to be busy in December, but if you have time and want to swing by Stanford and give a talk here, we'd love to host you.
For sure. Thank you.
Yeah, just some different types of questions. I've always heard that kids playing in the soil, it's important for them. And I'm wondering if anybody's done a study between the soil microbiome and the human microbiome, like kids eating dirt. Is that something you've thought about, or do you know about? Yeah.
Yeah, there was a paper. So I taught for many years at WPI, the anatomy and physiology course. And there was a paper I used to teach on microbiomes that was able to map some of the gut microbiome to some of the raw foods that were consumed in those different communities. So there were communities in Western Europe versus communities in Africa. And so I think there is a rich literature there. I'm not as familiar with it, but I've heard of examples. So for sure, I think it makes a difference, and many, certainly many studies that have looked at the effects of diet on microbiome. Yeah.
Another thing I was wondering about is just from the perspective of engineering and tools. Obviously, you're making these amazing advances given the tools that exist. First part of the question is like, what's been the most important tool in your toolkit? And then what's your greatest frustration in terms of a missing tool? If you could snap your fingers and some new magical tool existed that would let you do something that isn't now possible, what would you wish for as your next gen toolkit?
Oh, that's such a good question. I wish that I could monitor the microbiome composition in real time over time. I wish I could watch it evolve, because right now the best we can do is take snapshots, and it's biased by the efficiency of the DNA extractions, and and that's the best we can do. But I would just love to just be able to watch it evolve, watch the interactions happen. I would love that. I'm not even sure what kind of technology you would need to be able to do that, but that would be sort of my pipe dream tool that I'd love to have. I mean, that being said, I think for us the key was looking beyond. So finding other people who are doing stuff on mostly like cattle grazing lands is where the Rosal lab is. They're not a synthetic biology lab. They were developing these soil models so that they could measure the effect of livestock on the microbiome, and there's more microbial ecology based people. So for me, getting inspiration from people who think about dirt every day from other points of view has been really critical.
Yeah, empathy for dirt.
Yeah, absolutely.
What if it's 20 years from now and everything, like let's say you can see what the microbes doing their thing, and everything you wish for comes true. Like we understand the soil microbiome, we can partner with it as you wish, right? Like, what do you think that world's like? What's that going to be? What are we going to be able to do, or how do we see things differently because of that? When your nerd rapture is real, like, what's that world look like?
Yeah. Well, I think it means that we can reclaim a lot of the earth that we've given up on, right? And back to your first, when you were speaking at the beginning and you had this great comment about how do we get to a place where we can coexist with this earth without trashing it. Some of that is going to require undoing what's been done and reclaiming that. And I don't think that can be done for the soil until we really have this understanding, and then also understand how to work with it, as you just said. So once we understand how we can intervene with an engineered organism, and then how do we get rid of that organism, and how is that organism going to affect the broader microbiome and the broader landscape, and the interactions with the plants and the interactions with the animals and the animals' gut microbiomes, right? Until we have that big picture, it's going to be really hard to reclaim land and undo what we've done. But I think that that's key to getting to nerdvana, right?
Yeah, I really what you're saying really resonates with me. I love your future. Thank you so much for your talk. I'm going to excuse myself. I hope we can get you to visit in December when you're out in California.
Yes,
Fantastic.
absolutely. I'll be in touch. Thank you.
Thank you so much. Thank you for your time. So I have some more question.
Okay.
Let's see. the also final Drew's question. I will read this one. What do most people not understand about soil microbiome? What's the most important dirty secret hiding in plain sight?
Dirty secret hiding in plain sight.
Yeah.
That's a good question. What don't people understand?
Yeah.
one thing that I read that really sort of blew my mind, it almost gave me like a moment of hopelessness, but then I sort of bounced back from it. But there have been studies that show that even, if you take you can take a gram of soil and you can extract all the DNA and you get a microbiome, right? So these are the members, this is who's here and in what ratios. But if you look even smaller, those microbiomes, there are subbiomes within a biome that exist on individual grains of organic and inorganic material. So if you look even smaller, the microbiome that might be on this piece of sand, which is also part of this same gram of soil as this stick over here, are different. And so, what do you do with that, right?
Yeah.
So when you extract a gram of soil and you extract the microbiome, what you don't see are the subbiomes.
Right.
And those are really different depending on if there's a root nearby, right? Then you have a rhizobial community that you're having to capture. And if there's organic content that's up against mineral content, then you're going to have different. So I think that it's just, we understand, okay, it's complex, but I think it's even more complex than we like to think it is. And I almost don't know how to deal with that complexity, but that's what I would say.
Yeah, I would say you are so brave enough. And then it took me more than 10 years to jump into the soil, but you already, jump into, when you are young, so that's fantastic. So I have the question, from the audience. So Maya asked, what are the effects of the gene circuit on survival and competition? And do you have any thought on how to account for survival competition when designing gene circuit?
Yeah, that's a great question. So right now, we don't think that, so the gene circuit that we're using in the CSOM experiments is just expressing TD tomato. We don't have any evidence that that's causing anything like a significant metabolic load or anything like that. So we're using a system where there's a single copy integration of that fluorescent protein. So it's not a big burden that's affecting survival, we don't think. Ideally then, what you would want to do is put in a gene circuit that does a thing. So we haven't done that yet, but you'd want it to sense something or break something down or remediate something, right? And so we're building those circuits now, but we haven't tested them in the CSOM yet. So we don't know what the answer to that is just yet.
Okay. All right. So another question from somebody. You see that P. putida dies off in the non-sterile soil. How different do you think the survival would look if you had isolated a microbe from the soil you make the extract from and engineer it and then put it back in the original environment? That's very interesting.
Yes. Well, and that's definitely an approach that a lot of folks in this space are taking. And for good reason, right? So there's some logic to taking an organism that is going to do well in a given environment and then engineering it to have your function. So the barrier to that is the tools for engineering in these strains. And some of the most dominant soil taxa are not culturable. And or even if they are culturable, there are no genetic tools that are developed. They can't transform them. You can sometimes get genes to go into them through conjugation and things like that, but it's labor intensive. And so, but that's definitely an approach. And some folks are taking that approach. We're sort of taking the approach of, if we have something that's easy to engineer and that is a member of the soil community, even if it isn't a dominant member, maybe it's more accessible for us to modulate its survival than it is to take an organism that's harder to engineer and put our gene circuit that we might want to run into that organism. So it's more of a philosophical difference of how you approach the problem. But absolutely, people are doing that, just as that was described.
Sure. Let me double check. So I guess we answered all the questions. now I see 11:20 Central Time, so I probably need to close. Okay, so we'll chat again, official, more casual way. So I'll close. So thank all for joining and staying today. We'll meet again on May 18th, Thursday, the same time, the same Zoom link. We'll have Professor Guibart Stan from Imperial College London and Dr. Alex Darlington from University of Warwick, both UK. As usual, the follow informal chat will occur without recording. Please stay here. If you are interested in chatting with us, I will promote you to panelists who can speak and show your handsome and pretty face if we wish. And thanks, I will stop recording. Just 1 second. One more.